Record. I'll make the pop-up go away.
That's okay, right?
Yeah, cool.
Yeah.
Hi.
Bring out the right signs, like killer.
Yeah, from the back. We're doing well. No, I
That's
do
right,
too.
yeah.
My community from that.
And you want?
It's like.
Yeah.
Hi. Hi, James. Hi.
What do you guys want?
Yeah.
That could be good.
I was gonna say.
Okay, yeah. Yeah.
We'll have the round two. No.
Yes.
So. Yeah.
It's so weird how hard it is to turn this thing
That's it?
Yeah, I heard it.
Hi.
Is your name, is it Greek? I'm trying to figure out.
It's hard to say. Because my mom's cousin came up with it, but they're not sure like where he came up with it. But I think it's like it's inspired in Egyptian names. My mom's name is Isis. Yeah. And there's like someone named Nepsis in the mythology, so maybe like somewhere around there.
Really wonderful. The volume should be up here. All right, y'all. Thanks for joining us today to hear Nepsis talk about the research she will be conducting. Next, Nepsis comes to us with a BS from this name I've read many times, but I've never said it out loud until this moment. Ready? Pontificia Universidad Católica Madre y Maestra. That's the whole name of the university, right? So that's where she got her BS. She got an MS at Nova Southeast University. And so Nepsis is really interested in coral, as you all will learn. And I was really excited to bring her into the lab because she has this sort of she has this sort of enthusiasm and energy and optimism that I think is really required to study coral. As you all may know, coral are not doing so well. And what I really appreciate and respect and draw a lot of energy myself from is her sort of her energy and motivation to really understand what's driving these things and what we can actually do to make a difference, right? So it's very proactive as opposed to saying, oh, why are they getting degraded, right? And so I just really enjoyed having her in the lab, and I'm really excited about the work she's gonna be doing. You all get to hear about it now, and with that, I'd like to introduce Nepsis.
Thank you, Jay, for those kinds of words. Hi, everyone. Thank you so much for being here. I feel loved right now, so thank you very much. My name is Nepsis García. I'm a second-year PhD student in the Ecology lab, and today I will be presenting you my dissertation proposal on coral, on the mechanisms that coral use to maintain production. So one concept that is going to frame most of my dissertation is going to be fitness. And fitness is an organism's ability to be able to survive, grow, and reproduce in changing environments. And this fitness is actually underpinned by this other concept that is physiology. Physiology actually is all those processes that make this fitness possible by going through different pathways of metabolism and biochemical processes. Fitness is primarily important for ecosystem engineers because these organisms are the ones that will use and modify both biotic and abiotic resources in their system to be able to build habitats for other species. So we can use fitness of specifically ecosystem engineers to be able to understand the production of these spaces. Say, for example, coral reefs. Corals are the ecosystem engineers of coral reefs, and because they're able to build these strong structures by using all these resources, they're able to provide this habitat for different organisms by building these cracks and other different spaces where they can thrive, such as, for example, these fish, fungus, and others. But in this space, besides corals, we also, in organisms, we also have these environmental factors that are affecting this space, right? And all of them are happening actually at the same time. The changes in them are going to also be affecting their fitness. The problem with this is that we often study them in isolation and we study them in controlled environments. And I would know because I have been doing work like this myself for my master's. So because we study them in the lab and we study these factors and their effects in isolation, we lose a lot of information that is very important about how they interact and about how those interactions will change how there would be different responses in the organism. And this is why my research will explore the mechanisms underpinning coral fitness under multiple environmental factors in natural settings. Now, coral reefs are ecosystems that thrive in nutrient-poor environments, but paradoxically, they're one of the most productive systems on Earth. And this is because they're very efficient at recycling energy and nutrients. They also provide ecosystem services for both marine life and also humans, such as coastal protection, creation of habitat and maintenance of this habitat, fisheries, and others, while they also play a role in the uptake, fixation, storage, and release of carbon. But besides all these amazing things that coral reefs can do for Earth, they are experiencing severe declines due to different environmental stressors, most of them that are originated from anthropogenic activities. And they're being further exacerbated by the increased intensity and frequency of heat waves and hurricanes. Corals are complex organisms that are mostly known for being colonial, but the most simple form of their life is a polyp. A coral polyp, if we zoom into a coral polyp, we can see that they have these little freckles that give them color. Now, these freckles are actually unicellular dinoflagellates called zooxanthellae that have a symbiotic relationship with the animal. And in this relationship, the zooxanthellae photosynthesize, providing the coral with the byproducts of photosynthesis that include carbon, which gives the coral a source of energy, whereas the coral, by capturing food items from the water column, can provide both of the organisms with nutrients to be able to further fuel this photosynthesis. And this further allows the zooxanthellae to be able to fix biomass, and both the zooxanthellae and the corals fix biomass, grow, survive, and reproduce. There are 4 specific physiological processes that I'm very interested in that are happening in coral. The first one is photosynthesis, which we already talked about. It is basically that relationship that the symbiotic algae have with the coral, where they can fix biomass, grow, and survive. We also have calcification, which is the process by which the coral grabs calcium and carbonate from the water column, and they're able to build these calcium carbonate skeletons beneath their tissue, and which is what allows them to build these structures that form habitats for other organisms. We also have nutrient cycling, which is a very efficient and tightly knit process that happens between the coral and the zooxanthellae, where they are very efficient at maintaining these nutrients and energy because they live in nutrient-poor environments. And we also have gametogenesis, which is the process by which they create gametes. And this is particularly important for sexual reproduction, because through sexual reproduction is that corals are able to increase their genetic diversity and also have gene flow between different coral populations. Now, these processes are also affected by environmental factors. And there are 5 specific environmental factors I would be interested in for my dissertation, which are light, nutrients, pH, temperature, and food availability. Light directly affects photosynthesis. Nutrients directly affect photosynthesis, calcification, and nutrient cycling. pH directly affects calcification. Temperature affects gametogenesis and photosynthesis. And here I want to make a brief stop to talk a little bit about temperature. So I'm pretty sure that everyone in this room knows that temperature has been affecting ecological systems everywhere in the world, and coral reefs are no exception. When temperatures increase, the risk of coral bleaching increases as well. But what is coral bleaching? Coral bleaching is a process in which the coral actually expels the zooxanthellae from its tissue. And they look like this. That's why it's called coral bleaching. They look completely white, and this means that you're only seeing the color of their skeleton. Now, if they don't recover from this quickly, then they enter a phase of being so stressed physiologically that it can eventually end up in their death, especially because photosynthesis can provide them with approximately 95% of their energetic requirements. So they're losing all this energy from coral bleaching. Now, moving on, food availability affects nutrient cycling, photosynthesis, and gametogenesis directly. But we know that all these things affect these things directly, right? But they're all happening at the same time in the organism. And if we go further, we see that there are also indirect effects that these environmental factors are having in the organism. So within the context of the coral, a lot is happening at the same time. And this makes it pretty complicated for scientists to be able to detangle what is going on when one thing or the other is happening, or when one thing and the other are interacting. And this gets further complicated because if these environmental factors get outside of this optimal range that is good for corals, then they can also be detrimental for the coral and they become stressors. An example of this is a study by Fabricius et al. In 2011, where they looked at ranges of pH in which corals would do well. So on this graph, we can see that coral development, specifically in terms of hard coral juvenile density, was throughout this range of 7.8 and 8.1, pretty stable. But they also found that in places where pH was below 7.7, development completely stopped, which tells us that pH in this scenario went from being something that affects the coral to being something that stresses the coral specifically. Interactions between these stressors are often non-additive, meaning that the sum of the effects are not necessarily going to be what the effect itself is going to be on the coral. And when these environmental factors change, the effects of them on the organisms can often be cryptic. A good example of this is a study that was conducted by García et al. That was published in 2020. In this experiment, this experiment was conducted in the Bahamas for 10 months. And they were manipulating the fish-mediated and anthropogenic-mediated nutrients. To be able to assess how the corals will respond to these different treatments, they assessed coral growth, zooxanthellae density, and they also assessed 7 different physiological variables, including stoichiometric variables and also stable isotopic variables. And to be able to analyze the results, they use structural equation models and ANOVAs. We'll get into that a little bit later. Now, something very interesting that they found in the results was, well, first I want to make you aware that each tone of green is a different coral species that they evaluated. And then here we have the different treatments, which will be a combination of fish-derived nutrients and fish-derived and human nutrients. What they found here by looking at the coral's growth was that within each species, there was no significant difference between the different treatments. Now, I want you to remember that growth is a basic or traditional measurement of the fitness of an organism. Now, where this study gets interesting is when they take a deep dive and look into the coral's physiology. Now, I'll make a little stop here for a second and talk a little bit about structural equation models. Structural equation models are a tool that were used in this study, and they're very useful because they can help you understand complex interaction between different variables. They allow you to use response variables as predictor variables, but other response variables, they also allow you to measure the direction of the effect of one variable on the other, and they also allow you to measure the magnitude of their effects. So on the fish mediated nutrient treatment, they found that there was a pretty even distribution between what was dominating the exchange of energy and nutrients within the coral, between the coral and the symbiont. But then when they looked into the treatment that introduced anthropogenic nutrients, they realized that there was a shift in the dominance of these exchanges. And it moved to being more dominated by the symbiont or the zooxanthellae. The authors suggested that they might have identified a point in the coral that was right before this shift happened, where the mutualistic relationship between the two organisms was moving from being a mutualistic one to an antagonistic one. This lets us know that there was an environmental change that had a cryptic effect on the coral fitness because they were not able to detect it in growth, but they were able to detect it in the physiology. This is a critical detail that they would not have detected if they only relied on traditional fitness measurements. And this is why it is imperative to have a comprehensive physiological approach to be able to answer these complex questions where a lot of things are interacting at the same time. Another example of this is this case study by Wright et al. In 2019. In this study, they looked at 40 genotypes of the same coral species, and they measure a variety of different physiological variables and also fitness variables. They also controlled for 3 different stressors at this, separately and also combined. And what I want you to take out of this figure of this study is that they did not see any significant changes across all the different levels of treatment or the combined one, which led them think that there's no direct negative trade-off when they have one versus multiple stressors at the same time. But there is a caveat for this study, because the study was conducted in a control setting in a laboratory. So, and it was also conducted for only 10 days. So the things that all Gallardo into 2020 could have detected because they had this wide approach, because they did it in the field and they did it for 10 months. These people might have lost it because they only did it for 10 days. And this is why, for me, it is very important to be able to combine the ideas and of both of these studies to be able to address my dissertation. My dissertation will experimentally study the mechanisms underpinning coral production across a gradient of multiple anthropogenic stressors using fitness and physiological measures in real-world settings. I will be specifically addressing 3 different fundamental ecological questions. For my first chapter, I'll be answering how does light limitation interact with environmental variables to affect coral fitness and trophic niche. For my second chapter, I'll be addressing how genetic diversity and gene flow affect coral fitness within and across local coral populations experiencing different environmental conditions. And for my third chapter, I will be addressing how does habitat architecture influence community composition and nutrient supply, and how do they mediate coral production. So for my first chapter, where I will be addressing how light limitation affects fitness and trophic niche, first I want to give you a little bit of context. So light limitation, when light limitation happens to corals, they shift the pathways in which they acquire carbon to be able to supplement their energetic requirements. And there are 3 different adaptations that have been found that they use to be able to deal with these conditions. One of them is changing their symbiont composition within the host, which is also called symbiont shuffling. And what they do is that they expel the symbiotic algae that are within their tissues and grab other symbiotic algae from the water column that hopefully will be different species that will be more efficient at photosynthesizing under these conditions. Another one is that the zooxanthellae themselves would be able to rearrange the light harvesting complexes within them to be able to be more efficient photosynthesizing. And there's a third one where the corals rely more on heterotrophic feeding to be able to acquire their energy. Now, corals are mixotrophic animals, which means that they can use different sources of feeding to be able to supplement their energy. We've already talked about autotrophy, but they can also do heterotrophy. This, a specific case of this is corals that live in the deep sea, because there's no light that reaches there, there cannot be photosynthesis, so they rely solely on heterotrophic feeding. An example of heterotrophic feeding being used for, in a stressful condition is a study by García et al. In 2020, where they use Bayesian mixing models to be able to understand how different coral species would prioritize either autotrophy, heterotrophy, or a mix of both of them. Now, what I want you to really focus on here is how they plotted the stable isotope for carbon and nitrogen for both the coral and the symbiont. So here, the coral host would be the lilac or purple dots and ellipses, and the symbiont will be the green dots or ellipses. Now, on the top left graph, what we can see is that there is a 94% overlap within the coral host and the zooxanthellae. And this lets us know that it is very likely that most of the energetic requirement for this coral species was being supplied by autotrophy. Whereas on the bottom left, we can see that there is a 0% overlap between the zooxanthellae and the coral host, telling us that it is very likely that these corals prioritize heterotrophy. Now, in this same study, they also wanted to identify how corals will respond to heat stress when they would prioritize one or the other. And on this graph, on the x-axis, we can see that there are days that went by where temperatures were different on the y-axis, but I want you to really focus on the vertical lines that have different colors for different species. Each of these vertical lines is the moment at which that coral species bleached. I want you to notice that the corals that prioritize autotrophy bleached pretty early in this range, from around 50 to 55 days after being exposed to high temperatures, Whereas the heterotrophic corals bleach a little later on this range. So in the study, they found that corals that prioritize heterotrophic feeding are more resilient to heat stress. So I'm wondering, what happens with light limitation? Would they have the same response? Would they use the same pathways to be able to deal with these stressors? And that is why for this chapter, I will experimentally test how gradient of light limitation influences coral fitness, both independently and interactively with other stressors. For this chapter, I will be studying the Samana Bay in the Dominican Republic. Now, the Samana Bay is a very interesting system. And that has two rivers that discharge within the bay. It also has a mangrove forest, and mangrove forests are widely known for being a nursery for zooplankton. And because of the river and the mangrove, there are sediments and there's also high densities of zooplankton that limit the light penetration into the bottom of the bay. And this creates a very strong light gradient in the bay, which I think is very interesting to study. Now, this is what's happening on this side, but on this side, we have the open ocean. So I'm interested in setting up 3 different sites in this light gradient to be able to identify how the corals would have different responses at these different places. So specifically to be able to do that, I will be deploying as a part of a larger ecosystem scale experiment for my lab. We'll be deploying clusters of 9 artificial reefs at each of these sites, of which I will be using 4 of them, which are the ones colored in green, to transplant corals and be able to identify different dynamics at the reef level. To be able to do this, I will use rebar reef stars to be able to transplant corals. I will specifically use 3 different coral species, 4 fragments of each of them on these 4 artificial reefs at each site, for a total of 12 fragments per site. The response variables that I will be evaluating will be coral survival, growth, fecundity, zooxanthellae density, and trophic niche. I will also be evaluating the exchange of nutrients and energy between the coral host and the zooxanthellae, just like they did in the Algai et al. 2020 paper, to be able to understand if there is a shift in the interactions between the coral host and the symbiont. Further, I will also be analyzing the stable isotopes of carbon and nitrogen within the coral, the zooxanthellae, and the zooplankton to be able to see whether there's a change in the trophic niche as they go along the light limitation gradient. And finally, I will also be tracking all these environmental factors that I'm interested in: light, nutrients, pH, temperature, and food availability, to be able to understand how they might be affecting all these results. For this specific chapter, I will be sampling 3 times a year for a total of 2 years. So for this chapter, I hypothesize that as food availability increases, I will also be seeing an increase in coral fitness, regardless of light availability. And this is because when light availability increases, then food can also increase. And then when food increases, then light can decrease. So there's like a certain dynamic of nullifying each other. And that's why I think it will be, that's why I hypothesize this. I will be able to analyze this by using mixed effects model, which will allow me to do different iterations of models to be able to identify how each of the response variables will be affected by all the different environmental factors at different sites. I will be using AIC to be able to identify which of those models will best predict the outcome of these responses. I also hypothesize that stressor interactions will have a synergistic effect on coral fitness. So specifically, I expect heat stress, nutrient pollution, and light limitation to have the strongest effect in this chapter on the response of the corals. And I would expect that the effect of the stressor will be greater than the sum of their effects. I will also be evaluating this with a mixed effect model, and
Something important to also take into account here is that light, even if it's necessary for coral development, it also can be detrimental to coral fitness, especially when it is when it interacts with nutrient pollution or heat stress. I also hypothesize that corals will achieve the trophic niche depending on light availability or light limitation. Specifically, I would expect that the site that is closest to the source of light limitation will show a very strong overlap between the coral host of the zooplankton from the area, indicating that they rely heavily on heterotrophy. Whereas I expect the opposite to be happening at the site that is closest to the open ocean, where there's a stronger overlap between the zooxanthellae and the coral hosts. For my second chapter, I will be addressing the question, how do genetic diversity and gene flow affect coral fitness within and across local coral populations experiencing different environmental conditions? Genetic diversity is a key factor in maintaining population fitness in corals, and it enables their rapid evolutionary responses to disturbances. There have been previous studies that have found that corals that are locally bred often show high fitness due to different adaptations, but there have also been studies that have shown that this can be manipulated with reciprocal transplants. One study to exemplify this is a study by Kenkel et al. In 2013, where they found out that the coral species Brydis that originated in environments with variable temperatures would have a higher resistance to coral bleaching. Whereas a study by Palumbi et al. In 2014 found that when they transplanted corals from a reef to a different place that had more variability in their environmental factors, then they would also show that they would have a higher resistance to stressors. And specifically in this study, they saw a long-term effect where after 2 years, these corals were still having higher resilience to stressors. Now, when we talk about gene flow in coral populations, we need to talk about sexual reproduction. So what you're seeing in this video is what is called coral spawning, which is the way in which corals sexually reproduce. Those little dots that are white, it's the gametes that are being released by this coral thicket. And it looks like it's snowing from bottom up, which is amazing. But anyways, gene flow in coral populations happens when corals release either gametes or larvae that can be transported in oceanic currents. And through dispersal, they can send these gametes or larvae to different parts, to different places where they can integrate into those populations. Now, when they integrate into these populations, they increase the connectivity between them. And this connectivity allows coral populations to be more resistant to different disturbances, because if one population gets wiped out, then it can be replenished by those other populations that send them this genetic material through gene flow. Because of this, understanding how populations connect and mix provides insight into the genetic differences between populations, and identifying the distinct management units is critical to be able to preserve biodiversity. Information on gene flow helps develop effective conservation strategies. And this is why in this chapter, I will evaluate environmental factors and population genetics within and across local populations around the Dominican Republic to be able to analyze how independently and interactively these influence population level fitness. I will also be conducting this experiment in the Dominican Republic, but it will be wider. I will be studying a local area where I will be looking into differences within populations to be able to identify whether there are multiple discrete populations, or if it is more of a one big discrete population. And then I will be interested in looking at how there's a connection between all these other 8 different localities around the country, to be able to understand cross-population differences. For this chapter, I will be studying 3 different coral species that are common in the Dominican Republic. My response variables for this chapter will be coral fecundity and zooxanthellae density. And for my genetic analysis, I'll be extracting tissue samples to be able to extract their DNA and then sequence it using reduced representation sequencing or RAD-seq. I will also be implementing the same approach that the Allgaier et al. 2020 paper was implementing to be able to understand different relationships within the coral host with the zooxanthellae, by including the elemental analysis of CN and P, and also the stable isotope analysis of both carbon and nitrogen. And I will also be tracking all the environmental factors I'm interested in. Something very interesting about this chapter, or at least that I like very much, is that I will not only be looking at how their genetic makeup is gonna change coral fitness, but I will also be looking into environmental factors that will be affecting them. So even if I can encounter some challenges with one or the other, I will still have kind of a very complex globalized chapter for this purpose. Something important that it's also relevant for the study is that I will be doing one-time sample across all these sites to be able to understand these things. It will not be a continuous sampling process. One of the things that I hypothesize I will find here is that the increase in genetic diversity and the increase in gene flow will affect coral fitness positively, either if both of them are high or just one of them is high. And this is because genetic diversity is very important for coral populations' resilience to environmental factors, because it makes it gives them more strength to be able to withstand these processes. And gene flow allows for genetic diversity to be increased in these populations. To be able to analyze these effects, I will also be using a mixed effects models approach. Another thing that I hypothesize is that coral individuals with similar allelic frequencies to conspecifics at the same site will show higher fitness than those that are more dissimilar. And I expect to be able to see this by using clustering assignment methods, where I am hoping to see that there would be a clear separation between corals that are similar and have low fitness, corals that are similar and have high fitness, those that are dissimilar and have low fitness, and dissimilar and have high fitness. And I expect this because higher allele frequency similarities often indicate more gene flow, and this enhances coral adaptation to local conditions, whereas in contrast, dissimilar corals may adapt to different environments, and this can increase the risk of reduced fitness due to maladaptation. The last thing that I hypothesize that will happen with this chapter is that there will be high levels of connectivity within populations, but low connectivity across populations. So I am expecting to see isolation by distance, which establishes that the more geographic distance there is, there's more genetic distance there's going to be. But I also expect that these coral populations will encounter environmental barriers that will further increase that distance, that genetic distance that will happen the longest they get separated from one population from the other one. Now, for my last and third chapter, I will be addressing the question, how does habitat architecture influence community composition and nutrient supply, and how do they mediate coral production? Now, it has been for a long time an ecological goal to be able to understand the primary factors that underpin ecosystem structure and function. And this is often framed within top-down and bottom-up forces, which basically establish that either higher levels of a trophic web versus lower levels of a trophic web will have a stronger effect on each other. But looking at this from this perspective can be unrealistic, because these ecological systems are so complex, especially when we're talking about coral reefs. So assuming that the response is going to be a binary one is not very intuitive in this case. And that is why ecologists have decided to have a more nuanced approach to this. And look at this framework from the point of view of them being two simultaneous processes that are happening and that can integrate with each other and interact with each other. One example of this is a diagram that I created here to conceptually explain these interactions. So here we have different players that are within a coral reef. We have predators, omnivores, coral, algae, nutrients, all these different players that have not only arrows that go from the bottom up, but also arrows that go from the top down. And depending on the combinations of these different variables, we can see that there are different results that can be either positive or negative. Now, to be able to study bottom up and top down controls, a good way to do this is by looking into habitat architecture. Habitat architecture is the size, shape, quantity, and arrangement of spaces and structures that organisms utilize. And the habitat architecture has been found to affect fish and benthic density and community composition, and has also been seen to influence top-down processes through direct predation and direct behavioral impacts and bottom-up effects. A very interesting way to look into how habitat architecture can change these different processes is by using artificial reefs, which are like the golden child of my lab. So artificial reefs are super interesting because they are discrete experimental units that you can utilize to study different reef level dynamics. They offer three-dimensional structures. They offer three-dimensional structures that can offer a lot of structural complexity, creating a lot of spaces and cracks for things to colonize or habitate, especially fish. A lot of fish aggregations increase a lot when we deploy artificial reefs because there are a lot of spaces where they can find refuge. And this increases fish aggregations, which also increases nutrient availability, creating hotspots in these places. But artificial reefs also offer a lot of substrate for benthic organisms that can colonize them, which is what makes these units very interesting to be able to study top-down and bottom-up dynamics. And this is why for this chapter, I will leverage a long-term artificial reef experimental project in the Bahamas to quantify how nutrient supply, benthic, coral, and fish community composition mediate coral production. Now, these artificial reefs have been built as clusters of 9 artificial reefs in the biome of Robin, specifically in Abaco Island. And they have different distances between the artificial reefs. This one is of 1 meter, this one's of 3 meters, and this one's of 5 meters. And this was built this way so that we could manipulate the different habitat architectures to be able to identify differences. I will be specifically studying 4 of those 9 artificial reefs at each cluster, which are the ones that are colored in pink. And this will leave me with a total of 12 artificial reefs that I will be analyzing for this study. My response variables will be proxies for coral production that will be specifically coral cover and coral recruitment. I will also be analyzing the different community compositions of fish, coral, and benthic organisms. And I will be interested in different aspects of the community composition of each of them. And I will also be tracking all the environmental factors that I am interested in for this chapter. I will be measuring all these things annually for a total of 3 years. Now for my methods, and I think this part is very cool, I will be using structural motion photogrammetry, which basically means that I will take a lot of high quality images that from different angles that will allow me to build 3-dimensional models of these structures that will not only give me the flat dimensions of the corals and everything that's growing on the artificial reef, but it will also give me the vertical relief of them growing vertically. And then I will use this tool called TagLab that will allow me to trace a polygon around each of these organisms and will automatically calculate the flat area of the organism, but also the surface area of the organism. I will also be conducting underwater visual fish surveys to be able to understand the fish communities within these artificial reefs. And by getting this data, I will also be able to estimate fish biomass. And from the biomass data, I will also be able to estimate fish-mediated nutrient supply. And finally, I will also be deploying 5 terracotta tiles on each of these artificial reefs to be able to track coral recruitment at the sites. So what I expect to find, or I hypothesize for this chapter is, first, that when there's less distance between the artificial reefs, so the green line will be the 1 meter distance, I expect to see an increase in fish biomass. And I feel pretty confident about this particular hypothesis because it is something that has been seen in previous previous fish surveys that have been conducted in my lab already. And this also makes me confident to think that I will also find that nutrient supply will increase with these reduced distance between them. And because nutrient supplies will increase, I also expect for benthic cover to increase and coral size as well. To be able to analyze this, I will be using a permanova. Now, for this chapter, I'm very excited to be using structural equation models as well, because it will allow me to analyze all the different interactions that could happen within all these actors in the system. I will be walking you through different things that I hypothesize will happen within this project. First, I expect that fish biomass will increase nutrient supply, and that nutrient supply will increase CCA, which is encrusting calcareous algae that has been found for multiple of their species anyway to induce coral recruitment. I will also expect nutrients to increase algae and to increase coral cover and coral recruitment. I also expect that predator fish will have a negative direct effect on omnivore fish and herbivore fish. And I expect herbivore fish to have a negative direct effect on algae, and herbivore fish to also have a positive direct effect on coral cover and coral recruitment. Whereas omnivore fish will have a negative effect on aggressive invertebrates, coral cover, and coral recruitment. I also expect algae to have a negative effect on coral cover and coral recruitment, and also aggressive invertebrates having a negative effect on coral cover and recruitment. CCA to have a positive effect on coral recruitment having a positive effect on coral cover, and coral cover having an indirect positive effect on coral recruitment, because the presence of conspecifics is also something that induces coral settlement and recruitment. To understand the mechanisms by which corals maintain production in the Anthropocene, we must study them in their natural setting. I hope that as a result, my research will inform stakeholders on managing local stressors to be able to alleviate global ones, improve spatial planning strategies by protecting coral reef communities that are highly connected, and enhance our understanding of the potential of artificial reefs to be used as noble habitats to restore coral communities in coastal ecosystems. And with that, this research will provide a foundation of informed science-based strategies to safeguard coral resilience against increasing environmental pressures. I would like to thank my funding sources for funding my research. I would also like to thank my amazing lab. First and foremost, Jake, which is my advisor, for being so good at giving me advice and mentoring in the best way possible. Also, my entire lab for always giving me good feedback and comments to make me a better scientist, as well as my committee for doing that as well. I want to give a special thanks to my family, first and foremost, my wife, Carla, for being so amazing at making sure that I keep hydrated and fed, but also helping me make these beautiful slides and helping me practice and listen to me a million times, as she said, practicing this talk. Also, my beautiful cats that always help me stay grounded and present when I need it the most. My friends, especially Pad. Pad has been amazing to me all the time. Thank you so much for always being available to answer my questions, even if it's in the middle of the night when I'm freaking out about prelims. And Cheyenne for being my #1 cheerleader, always telling me that I'm gonna do great. I would also like to thank Ellie and Courtney, which are not in pictures here, but they're here in spirit. They are two undergrads that are helping me to process data from my research. So thank you so much for all their help. And with that, I will take any questions.
Love it. Okay. Hi, Nepsis. First of all, great job. So proud of you.
Thank you, Jay.
We're so excited to see this happen. I love all of your designs. So I am just a humble molecular evolutionary biologist. I don't do ecology, so all of your super wonderful complex models, too much for me to think about, right? So I have a few questions and they're all about chapter 2, because I feel much more at home there.
Okay.
So first question is, you're gonna do a bunch of sequencing and you're gonna do a bunch of population genetics to understand population connectivity, demographics, things like that, but also you kind of have this underpinning concept of fitness, right?
Yes.
So I want to ask, in the coral world, do you guys have an idea of genes and loci that are important for fitness? And is that going to inform kind of, because that's how you're going to do rad-seq, right? So
Yes.
are you trying to sequence and do things with kind of whole genomic data, or do you have particular loci of interest that you care about, that you already know about going in?
Yeah, that's a very good question. Initially, we were thinking about doing whole genome sequencing for this project, but that, as you probably know very well, can be pretty taxing economically. So we were trying to find an approach where we could encompass a wider area to be able to have like more interesting results without, breaking the bank. And I talked to a couple experts, and there are different genes and loci that we can look into to potentially be able to answer these questions. But more importantly, I'm interested in looking at how the genetic diversity and just gene flow in general, if there is any correlation with the coral fitness. So I can arrive there from different pathways. Does that make sense?
Yeah, for sure. And the last thing that I'll say, and we'll have time to chat about all these things.
Yes.
One thing that you could do, right, to kind of get at that question, there's like two approaches, right? There's the, I know what genes I care about, so let me just look at those, versus the, I'm not quite sure, so I'm gonna go see what comes out of it, right?
Yes.
And both are good. And you were talking about kind of bridging the value of in field versus in lab studies, right?
Yes.
You could, and this might be done, right? Because again, I don't work with coral, so I have no clue what's happened. You could do gene expression studies in lab, right? So you could just take coral species of interest, expose them to different combinations of environmental stressors, and take different time point sequencing measurements using RNA-seq, ATAC-seq, things like that,
Yes.
to see what genes and their expression levels get impacted at these different stages, right? So when a coral gets bleached, what's happening expressionally there, right?
Yes.
And maybe that's been done. But that's kind of the approach of, let's find out what genes matter.
Yes, yeah, and there has been, there's definitely a long way to go with corals being studied at a genetic level, but there have been studies that have looked into this as well. But more importantly, I'm more interested in seeing how they are doing, but how they're doing in their natural environment.
Cool.
Thank you.
So kind of wrapped up in your details of all the variables, and you're going to study in the ways in which you organize your chapters, which was very impressive. That I missed a little bit. What's the nature of your sample sites, and how much variation is there in those 5 environmental variables that you propose to monitor? And are you having replicate sites? So I'm curious about some of the outdoor attributes of what your study system will be like.
Just to confirm, are you referring to all of my chapters or one in
One and
specifically?
3 in particular.
Okay, okay, so to see if I understand you correctly,
Yeah.
you're asking how much spatial replication I will have?
Yeah, are you having for example, are you just studying one site at a time? Or do you have, say, several sites where you're monitoring all of these? Because if you're trying to look at how the environmental variables, the variation in those
Yes,
is interacting, then you're
Yes.
going to need either you're manipulating those or you're measuring what's already present in the natural environment.
Yes.
Or you have replicate sites or some combination of.
yes,
I'm curious
so.
How all that's working.
Yeah, that's a good question. Thank you. Yeah, I will have replicate sites and I will also be measuring all of them at the same time. Particularly for chapter 1, because the sites are along a very strong light limitation gradient. That will be like my central focus to be able to see how when these other environmental factors come along, how they change their response in the corals. Back.
Nepsis is a very cool project, very much needed.
Thank you.
Super stoked to see what are the results going to be. I was just asked, I was thinking about, and I'm pretty sure you are very aware of this. A lot of the coral reefs in the Dominican Republic are planted. So they are going to be clones of each other. And I'm thinking, is it something that you know how you're going to deal with it in the terms of the genetic diversity? Like, there's not gonna be much genetic diversity, they're just like sampling
Yes.
clones of each other in different places.
Yes.
So what are you doing to tackle that?
Yeah, that is a very good question. Thank you. Yeah, I've thought about clonality as well, because I feel like that's gonna be a big thing. I don't know if in terms of transplantation is gonna be a huge thing, because the DR is very big on transplanting the prettier corals like Acropora palmata or Acropora cervicornis. And I'm not focusing on those species just because they're not found a lot, right, in natural reefs. They're mostly have been transplanted, or they don't exist at all. They have been wiped out. Well, that is one way, right? I'm going to be looking at coral species that are not commonly transplanted in these areas. But also, there are ways in which I can tell if I'm running into clonality by using different genetic tests that I'll keep into consideration. But that is a very, very good question. Thank you very much. Yes. Yes. Oh.
He's not giving it up.
First, great talk. I love your spawning video.
Thank you. I love coral spawning.
For your first chapter, you said you're working with 5 species. What species are you?
Oh, I'm working with 3 species for the first one.
Which?
Yeah, that is a very good question as well. So going off of what Pat was saying, coral reefs in the DR are not doing well. There is a lot of death. There has been a lot of degradation on them. But we do know that there are corals out there. So I'm being flexible with what species I'm going to study, depending on what is most common in the bay, so that I'm not, like, if there are like 3 different colonies of this one species in the bay, that I'm not, taking tissue off of the only 3 the species that are there, and also because of representativity, right? So I'm sorry, representation. So I'm planning to scout the area to be able to see what species are more common, and then I will make a decision on which ones I'm gonna be able to study.
If you know the symbiotic algae they have.
Sorry.
Species have different symbiotic algae, and maybe you're gonna have different results in this fitness depending on the type of or species.
Yes, that is a very good point. That is something that I could definitely run into. But I will not be testing the different species of symbiont that will be within each coral. Primarily because my dissertation is already very expanded and complex. And getting into symbionts is a whole other universe. It is things that people focus their entire dissertations on. But it is definitely something very cool that I think about. Yeah.
I was curious, yeah, that was an excellent talk.
Thank you.
I really enjoyed that. I was curious, in chapter 1, your second hypothesis was that the stressor interactions will have non-additive effects on fitness. And I was just curious to hear more about the logic on why you think it would be non-additive or synergistic.
Yeah, that is a very good question. Well, we have found, well, there are studies that have shown that some of these, specifically the terms that I presented that would have this synergistic dynamic, we've seen them having different effects in combinations of two of them. For example, we know that when there's a lot of light, then temperature gets exacerbated and then thermal stress gets exacerbated. We've also noticed that when there's a lot of nutrient pollution, then when a heat wave hits, then the coral is more vulnerable, and then it will get their fitness will be further affected. So we have an idea of how combinations of two of these interact. So that is why I hypothesize that when they're all combined, they'll have an even bigger effects.
Great job, Nepsis.
Thank you.
I'm gonna follow up on a prior question.
Yes.
In your first and third chapter, your reefs seem relatively close together. So I'm wondering how much you expect those other environmental variables to vary
Yes.
in your different reefs.
Yeah, that is a very good question. Thank you. So some of the environmental variables, I don't expect to change as much from site to site because as you mentioned, they're very close by. But I still want to be able to account for them just in case there are some differences and there are some effects of them in the response of the coral. So that is a very good point. Yes.
There are no more questions. Something on Zoom. Let's give Nepsis another round of applause.
Like this.
Oh my God, you're so smart, Nepsis. You're so good.
That's great. I just wanted to.